What about the PCR, RT-PCR, qPCR, and RT-qPCR? Polymerase chain reaction, reverse transcription PCR, quantitative PCR, and RT-qPCR. كل هذه التقنيات الحديثة والهامة جدا في مجالنا، سواء علوم المولكولر أو علوم الجيناتكس والعلوم الفرعية التابعة لها أو علوم البايوانفورماتكس، وكل العلوم الحديثة في العلوم البيولوجية والبايوكيميائية. الحقيقة لابد من دراسة هذه التقنيات المختلفة. فإن شاء الله بإذن الله مع بعض يعني نحاول نلقي الضوء بشكل أكثر دقة على هذه التكنيكس بحيث أنها تفيد الباحثين والدارسين إن شاء الله. ونبدأ مع بعض بسم الله الرحمن الرحيم. What is RT-PCR? RT-PCR or reverse transcription PCR comprises two steps. The first one is the reverse transcription process, and the second is amplification of the desired DNA sequence by polymerase chain reaction. So in RT-PCR, RNA is used as a template. It is used to analyze and amplify a short stretch of RNA. RNA is the first converted to complementary DNA or cDNA by reverse transcription, followed by the standard PCR method. In RT-qPCR, RNA transcripts are quantified by the first performing reverse transcription, and then the standard qPCR is carried out. يعني في البداية بنستخدم ال RNA، هو بيكون المادة الأساسية اللي بيستخدم في عملية التحليل وعملية الامبليفيكيشن of PCR. RNA بيتم الأول تحويله إلى DNA عن طريق reverse transcriptase، وفي الحالة دي بيسمى cDNA. وبعد كده بنطبق عليه قياس هذه النسخ من ال RNA. بعد ما استخدمنا ال RT-PCR بنستخدم ال qPCR لقياس كميته. RT-PCR is used to detect the RNA in a sample, and the amount of RNA can be measured by using RT-qPCR. RT-PCR combined with the qPCR in the process or technique called RT-qPCR, which is very useful in doing quantitative analysis of viral RNA and gene expression. ويعني هيتم بقى القياس لل RNA باستخدام ال RT-qPCR زي ما قلنا، وده مهم جدا في عملية التحليل الكمي للحمض النووي الريبوزي الفيروسي، وأيضا لعملية الجين اكسبريشن. The steps of RT-PCR are the same as PCR with the additional first step of reverse transcription. يعني نفس العملية، لكن بتبقى في خطوة في البداية هتبقى باستخدام الانزيم reverse transcriptase، ودي هنبقى نشرح طبعا الخطوات دي بالتفصيل في السلايد القادمة إن شاء الله. In RT-PCR, complementary DNA cDNA is produced firstly by using reverse transcriptase RT. The cDNA thus formed is then used as a template for the standard amplification process by PCR. In more concerning with the choosing the total RNA versus mRNA, when designing a RT-qPCR assay, it is important to decide whether to use total RNA or mRNA as a template for reverse transcription. However, messenger RNA may provide slightly more sensitivity, but the total RNA is often used. This is because it has important advantage over messenger RNA as a starting material. وبالتالي اختيارنا للماسنجر RNA يا إما أو التوتال RNA بشكل عام بيبقى له هنا بعض الحيثيات والمعايير للاختيار، حيث أن غالبا ما بيتم الاختيار للتوتال RNA عن الماسنجر RNA فقط كتمبلت لعملية RT-PCR، وخاصة في الqPCR وعملية التقدير الكمي، ودي بتعتبر أحد التميزات، وبيبقى يعني اختيارنا للتوتال RNA بيكون أفضل في هذه التقنية. To sum up, RT-PCR and qPCR are the advanced methods of PCR. التقنيات حديثة فعلا ودورها هام زي ما هنشوف إن شاء الله. qPCR gives faster, more detailed, real-time results and is used to quantify nucleic acid, whilst the RT-PCR is used to detect and amplify cDNA and diagnosis such as طبعا كلنا عارفين إنه من أهم التقنيات اللي كانت بتستخدم في diagnosis الخاص بسارس كوف 2، هذا الفيروس المسبب لكورونا فيروسز بشكل عام. زي ما هناخد فكرة برضو عن هذه التقنية في هذا الفيديو إن شاء الله. Now we will study together the difference between PCR and qPCR in a simplified and more profound way, as we differentiate between them to carry out that aim of the qPCR in comparison with traditional PCR. The comparison will completed between me and Dr. Nelly in molecular biology. Number one, PCR is used to analyze a short strand of DNA by amplification.
1. It is an advanced PCR method, which is used to amplify as well as quantify the amount of DNA.
Number two, in PCR, primer is used for polymerization.
2. In qPCR, primer, as well as fluorescent probes or dyes are used.
Number three, results are investigated by gel electrophoresis.
3. Fluorescence emitted by dye or probe is recorded during the PCR process.
Number four, the data is recorded at the end of the process.
4. The data is recorded during the amplification process at the exponential phase.
Number five, ethidium bromide is used to stain the DNA fragments.
5. Fluorescent dyes or DNA probes labeled with a fluorescent reporter are used.
Number six, it is a low resolution technique.
6. It is a high resolution technique.
Number seven, distinct bands of various DNA fragments can be seen on agarose gel.
7. Different peaks related to different DNA fragments are seen during qPCR.
Number 8. Conventional PCR takes more time to generate a result.
8. qPCR takes less time to deliver the result.
Number 9. It is used to detect the presence or absence of DNA in a sample, detect gene mutations, and amplify the template for DNA sequencing.
9. It is used to quantify the DNA present in the sample. It is used to analyze gene expression, identification and detection of pathogens, quantification and identification of mutation.
Thanks, Dr. Nelly. Real-time fluorescent reverse transcription polymerase chain reaction. One of the most important applications for identifying and detecting virus infections, such as a virus SARS-CoV-2. هذه التقنية المسماة ب Real-Time Fluorescent RT-PCR اللي تعتبر أهم وأحد التطبيقات للتعرف والكشف عن الإصابات بالفيروسات مثل الفيروس الشهير SARS-CoV-2. نشوف الخطوات لتطبيق هذه التقنية الخاصة بال RT-PCR Fluorescent. الحقيقة في كيتس بتبقى معدة لديتكتنج بعض أنواع أو الكثير الحقيقة من الفيروسز للكشف عنها بتقنية ال RT. طبعا الفيروسز خاصة اللي هي المين جينيتيك ماتيريال خاص بيها بيكون ال RNA. فبناخد العينة من خلال السواب وبنحطها في اللايسيس بافر للحصول على الفيروس بشكل كمادة جينيتيك ماتيريال، ثم بنتعامل بها مع ال RT-PCR ماشين بالكلاسيكال فورم والتقنية السابق ذكرها بالتفصيل من خلال ال RT-PCR تيست. PCR master mix preparation and RT-PCR reverse transcription PCR. Preparation of the PCR master mix is a crucial part of molecular diagnostics, and it should be done in devoted room with dedicated equipment. So use clean disposable sleeves and gloves to avoid any risk of contamination, especially with DNA or RNA samples. And all stock solutions of reagents are stored at -20 Celsius to maintain this use in these diagnostic molecular experiments. So when you use all reagent should be allowed to thaw in ice except the enzymes reverse transcriptase and tag polymerase, where these enzymes it should be kept at -20 Celsius until used to avoid degradation. From this reagent stock solutions, primers, probes, it should be also prepared and stock this and stored these solutions in -20 Celsius. Avoid these reagents from to be pellet and materials not solution or not releasing. Please vortex and spin each reagent before use to avoid these pellets of material forming in the tube. Before the reaction mixtures is prepared, it's necessary to calculate the final amounts of each reagent required for the number of samples under examination. In addition of the positive PCR control and negative PCR control have to be considered in this calculation. In this session, one step of RT-PCR protocol is started. This protocol in a one-step reaction, reverse transcription of RNA and the amplification of DNA take place in the same tube. However, note that the reagents should be kept on ice for the duration of the master mix preparation. Prepare and label enough PCR tubes for the number of samples to be tested, and then separate the necessary amount into each single PCR tube. Then add PCR reagents as they are listed in the protocol. Distilled sterile RNase free water is used to dilute the reagents to be correct concentrations. And the tag polymerase buffer is needed to create the optimal chemical conditions for the tag activity. This important point. So add magnesium chloride is crucial for the activity of tag polymerase, as it acts as a catalyzer of enzyme reaction. Please note that in some cases it is included in the polymerase buffer, whereas the oxynucleotides ACGT are incorporated into the newly synthesized strand by tag polymerase. Besides that, the forward and reverse primers are short sequences of nucleotides and complementary to both strands of the double helix of the DNA that provide the initiation sites for the elongation of new strand of DNA by tag polymerase. RNase inhibitor is required to preserve the RNA template from the RNase degradation or digestion. For the preparation of the master mix for real-time PCR RT-PCR, a fluorescent probe is also added to the mix solution, such as the cyber or SYPR green is a fluorescent probe, is also added to the mix solutions of the this master mix real-time PCR RT-PCR and qPCR. This green or fluorescence cyber green is a free floating fluorescent dye that binds to the double-stranded DNA and increases in fluorescence concentration when bound. In qPCR or an RT-PCR, as the primers and the DNA polymerase duplicate the template strand, the cyber green binds to this double-stranded DNA as it forms. This cyber green quantitative PCR machines take readings of the amount of double-stranded DNA in each well at each cycle and give a critical threshold CT value, which represents the clarifying of your products, whereas CT is relative value. Whereas CT value is a critical threshold, which represents the clarifying of your DNA products which taking a fluorescent dyeing. And maybe CT is a relative value represent as showing in this figure in front of you. This is a short nucleotide sequence complementary to the target sequence, located between the primers that is linked to a fluorescent reporter dye such as cyber green. Please note that the props needed to be kept in the dark and away from the light sources to avoid reduction in the fluorescence dye. Finally, add the enzyme to the mix. This should be kept at minus 20 degrees until use. As we know that the TAG polymerase is a thermostable enzyme derived from the bacterium Thermus aquaticus, so it takes the abbreviation TAG. And it is able to recognize the primer template double. The TAG polymerase makes a complementary copy of the template by adding nucleotides to the primer. Whereas in this case of the steps of RT-PCR protocol, the reverse transcriptase enzyme is required to produce cDNA, complementary DNA, from RNA. When all reagents are included in the mix vortex, the tube to mix them and spin it briefly. Distribute the single reaction mixtures into each sterile PCR tube and keep the tubes on ice. If the mix contains fluorescent probes, remember that to keep the tubes in a dark to prevent the fluorescence degradation. The final PCR products of positive samples is called amplicon and consists of millions of copies of the target DNA. However, the amplicon is the expression meaning that a piece of DNA which of PCR product which amplified by this PCR and million copies of DNA or RNA. Put these tubes in the PCR thermocycler and program the correct parameters according to the used protocol of RT-PCR and start this reaction. Whereas this PCR, which is a sensitive machine, we're amplifying a specific DNA sequence according to the used protocol. When the run is finished, the PCR products can be easily visualized on the gel electrophoresis system using, as usual, UV light source. Note that the amplicon, which is a product of PCR, is stable for a few days at 4 degrees Celsius. Now the RT reverse transcription PCR is finished. Thanks for listening.